Hi, everybody. Welcome back. Um, in the last mini lecture, we started developing a modeling framework for physiological processes in a cell, focusing on mass action kinetics as the starting framework that we were going to use, seeing how we could develop simplified models for enzyme kinetics and build those up into models for a system of chemical reactions. For example, as you might see in a metabolic pathway or a signal transduction pathway. In this mini lecture, we're going to tackle a next major physiological process in the cell, that is the expression of proteins and the regulation of that expression. Let me start by switching over to the slides and moving from there. All right, so gene expression is much more mechanistically complicated than any sort of standard enzymatic mechanism. Part of the reason for this is that both transcription and translation, the two steps in expressing a gene into its final protein form, both of those steps actually involve many, many individual chemical reactions. Both steps require an initial assembly of an initiation complex where the primary engine that drives the reaction, RNA polymerase in transcription and ribosomes in translation, where those assemble onto the template, DNA in transcription, mRNA in translation. This assembly process can involve a lot of pieces, particularly in transcription, involves a large number of different components. Following assembly, there's the repeated extension of a polymer, a step, a chemical reaction that occurs over and over and over again for hundreds or even thousands of times. And in the context of translation, this extension itself involves a merging of an external component, a charge transfer RNA, which itself had to be assembled as part of that process. Then in both cases, there's also some complexity in termination. Determining how is termination recognized, how does the reaction stop, how does the template dissociate from the molecular machine that was driving the process. If we could imagine trying to build a detailed mechanistic model of this, where we keep track of each step, but it would be a tremendous mess even for the simplest system. And it's also not even that clear that we can do that for some of the processes, right? It's not as clear that the repeated extension of a polymer, both of these enzymes are precessive, the machine sits on the surface and moves forward along. Standard kinetic models might not work perfectly in that sort of context. This doesn't mean that we can't do anything, but it means that we need to be inspired by the ideas of mass action kinetics, inspired by the approach that we took in the previous mini lecture, rather than building a mechanistically exact model. The approximations to enzyme kinetics that we discussed in the last mini lecture yes, they were approximations, but they were approximations that you could derive mathematically from a detailed mechanism. You could write down the mechanism, you can write down the processes involved, and then under some certain very clear assumptions, you get those rate laws. Um, they're approximate, but they're grounded in a detailed mechanism, and the assumptions that go into their derivation are enumerated. We're not going to be able to do that the same way here. We are going to have approximate models where we're really grouping a lot of things together, even things that we don't perfectly understand, into very simplified models for a very complicated process. So how might we do this? What does it mean to be inspired by mass action kinetics? Well, our inspiration is going to come in the form of trying to write things in a rate law, just as we did for enzymatic kinetics, but to have all of the complexity of transcription and translation sort of captured in that sort of rate law. For each gene, we're really only going to track two things. We're not going to keep track of initiation complexes and the progression of a polymerase along a template. We are only going to keep track of the concentration of mRNA and of protein, and really we're only going to keep track of complete functional mRNA and of protein. We're kind of going to count a reaction as complete after an mRNA has been completely made and released, after a protein has completely been made, released, and in fact folded. That's all we're looking at, the final functional forms, and not trying to keep track of any of the intermediate steps. And for each of these species, we are fundamentally only going to have two rate laws that we are going to write down. One of them is going to be a rate law that describes synthesis, and the other is going to be one that describes degradation. Degradation isn't something that we really talked about very much. It's often sort of glossed over as an essential mechanism, part of the anabolic, the catabolic part of metabolism, the breaking down of molecules to make energy, also involves the breaking down of proteins and RNA back into their fundamental components so that they can be used as building blocks for other proteins and nucleic acids. This degradation step is actually essential. If you don't have degradation, then material can only accrue. You can only get bigger and bigger and bigger. There's a lot of problems with that in multicellular organisms, but even in unicellular organisms, it's problematic. It doesn't allow for control of growth based on energy levels. It also doesn't allow for consideration of the fact that molecules will spontaneously break down over time. RNA is not the most stable molecule in the world, neither are proteins. There's a natural degradation to them. There are organized degradation pathways inside every cell that help that degradation happen smoothly and in a way that doesn't mess up cellular function in an unintended way. So each gene, we're going to track RNA and protein concentration, and each of those molecules, we're going to describe a rate law that describes synthesis and a rate law that describes degradation. This basic framework can actually be extended to have a fair bit of detail in it. These rate laws could be quite complicated, involving lots of different pieces and being dependent on the concentration of many different species, if we wanted them to. They also can be quite simple, and our models that we're going to work with are going to be among the simplest that we can imagine, to give us a feel for what we can do. But I don't want you to come out of this thinking that this simple framework is impossible, is unable to capture complex behaviors. It can't capture all complex behaviors, but it can capture a fair bit of complexity. All right. Given this framework, how do we actually come up with such rate laws? Let's work through each of the pieces of this. First of all, degradation. Starting here because the model that we're going to work with is an incredibly simple one. And we can think about how this process might be modeled for degradation, for both protein and mRNA degradation. The simplest model for degradation is a unimolecular process. Degradation is something that happens in the physical world, in the chemical world, abiotically all the time. Radioactive decay is a degradation process. Chemicals like hydrogen peroxide spontaneously decay regularly. And in those simple physical and chemical decay processes, the rate is often a unimolecular process. We might write our reaction here as our molecule X, protein or an RNA, going to nothing. This is an important sort of nuance of the model. In general, we are going to be ignoring the background of a cell in our modeling. That is, we're not going to be keeping track of concentrations of free amino acids and free nucleotides. We could. It is possible to do that, and we can have a degradation model where we keep track of when a protein degrades, we keep track of all of the amino acids that were in there and how that contributes to the pool of amino acids in a cell. We can keep track of the degradation of an RNA and how that affects the concentration of building blocks in a cell. Our models that we work with in this class are not going to do that, and thus when a protein disappears, when a protein is degraded, when an mRNA is degraded within the context of the model, nothing reappears in its place. It apparently just vanishes. What actually happens is that the material flows into the pool of resources that we are not explicitly following in our models. Writing down how our model would treat this, our degradation just makes our protein disappear, makes our mRNA disappear. The rate of this reaction is simply proportional to the concentration of this. This is a model that comes from each molecule having an equal random chance of degrading. And so the overall rate of degradation, if you have more material, more of it will degrade. If you have less material, less of it will degrade, is basically the idea behind this. And so we might treat as an initial model a degradation process for protein and mRNA using this very simple unimolecular model. I want to be very clear about a very, very important point when we build models like this, especially for complicated cellular physiological systems. Assuming a simple model does not imply that we think the process follows a simple mechanism. It's often easier to add complexity to a model if we realize that there's something missing, that our model is unable to capture important behavior. We can look at what are the missing pieces and what complexity might we add to our model that might fix that, that might correct that. It's easier to do that than to start with an overly complex model, to have stuff and then to try to take pieces away, to simplify it down, to try to understand it better and to understand what's important. If we want to really understand what the essential features are, one very good strategy is to start with a very simple model and ask what features can we see and what features can't we? Then add some complexity and ask what more can we learn by adding that degree of complexity, and so on. Protein degradation is by far a simple process. It is not fundamentally unregulated. It is not fundamentally even random in any real way. But we might start with a simple model and ask, how far can we get using this simplest degradation model? Right. Okay. Translation. Translational regulation is often viewed, often presented as being less important than transcriptional regulation. This should actually say translation is generally viewed as kind of awkward wording in that sentence. I'm not sure how true that this is. I used to sort of be of that mindset. More and more is being learned all the time about how translation can be regulated. And there are a lot of important places where translational regulation is sort of essential. However, as a starting point, we might take the traditional view of translation being largely unregulated. And in that simplest model, we might further take the simplest model within an unregulated framework that says the rate of protein synthesis is directly proportional to the amount of mRNA that is present. We think about templated synthesis. We're starting out with a molecule of RNA. We're ending up with a molecule of mRNA plus a molecule of protein. A single reactant, if you will, gives a reaction whose rate is dependent on that. If we have more template, we can make more protein. There's a ton of assumptions in this. A major assumption in this is that ribosomes and all of the building blocks are not limiting. That what's limiting the rate of a reaction is the amount of template. And if we increase that, we'll increase the reaction rate. These may be reasonable approximations when the amount of mRNA is fairly low. It will break down at some point. At very large mRNA concentrations, you might run out of ribosomes, you might run out of tRNAs, and those might become limiting and slow down the reaction. These, as well as the lack of translational regulation, are in some sense clearly incorrect assumptions. But once again, that does not mean that this isn't a bad starting point. We can see how far we can get, what can we learn with this simplest possible model of translation. And because we know some of the incorrect assumptions that underlie this, we have ways that we can adjust this if we wanted to. This could be adjusted into having something that looked at more like a logistic model, or which has a lot of similarities to the Michaelis-Menten form, saturated dynamics, where yes, it increases linearly when it's low, but when the concentrations of mRNA get high enough, it maxes out at some maximal rate that corresponds to the availability of resources, for example. That's one correction we could make. We could build in regulation in various ways if we feel a need. But as a starting point, let's start with the philosophy that we want to start with as simple a model as possible and see what we can learn with this very simple model. Understanding that adding complexity is easy. Okay, now moving on to transcription. We know that transcriptional regulation is essential for many processes that affect the kind of cell fate decisions that we're particularly interested in. And really, it can't reasonably be ignored. Certainly, there are genes which are not controlled very much, and you can write down models for the transcription of a gene that isn't subject to any control. But if we did that, if we didn't have control at any level, if we don't have control at translation, we don't have control in transcription, we don't have any sort of control or regulation in degradation, the range of behaviors we would be able to see are very, very few and very uninteresting. And so from this simplest model perspective, we need to add some more complexity in at this point. And so we won't attempt to ignore transcriptional control. We know there's a lot of different types of transcriptional regulation. Each of these types of regulations we're going to need to treat separately. There will be relationships between them, but we should think about them one by one. We might think about, for example, a case where a gene is constitutively expressed. This means expressed constantly at essentially the same level all the time. We might talk about an obligate activator, a case where we need to have a particular transcription factor present in order to express a gene. We might be interested in looking at repression, a case where a transcription factor turns off expression of a gene. And there are many more mechanisms, particularly mechanisms where we have genes that are under the control of multiple transcription factors that we might be interested in exploring. In this mini lecture, we're going to look at the first three of these only. So let's first of all consider the constitutively active transcription case. This is a very, very straightforward one. Not much needs to go into discussing it. There certainly are some genes that have this behavior that are generally expressed at same level under almost all conditions. These are often referred to as housekeeping genes in cells. They express proteins that need to be around under all conditions. They need to be around at about the same level under all conditions. There's no point of regulating them in a dynamic way because that's not what is necessary for function. Constitutive activity can be modeled by just applying a constant rate of mRNA synthesis. We have a gene, the reaction we can think of. We have the gene for a molecule X as a template, so it's still present after the reaction and it produces another molecule. And the rate of that reaction will just be a constant, called mu of X, for the symbol we'll use for this. Again, I want to be clear, a simple model does not mean a simple mechanism. Constitutive activity may come from very elaborate control mechanisms. You can imagine there being many, many different components involved in ensuring that these housekeeping genes are regulated at a constant level. And if those mechanisms were what we were interested in, we would need to have those built into our model. But if we have genes that are involved in our process that we're interested in, but we're not interested in the regulation of those genes per se, it may be reasonable to ignore them and say these are constitutively active genes that we need to be expressed, but that we can model with a very simple approach for dealing with constitutive activity, constant synthesis rate. Okay. Now, what about activation? There are some genes that absolutely require the presence of a certain transcription factor in order for transcription to work. This is what we call an obligate activator. There are other kinds of activators that simply speed up the process, but in the absence of the activator, the gene can still be expressed at a lower rate. We're going to focus first on the obligate activator case, the case where when the gene is not present, when the transcription factor is not present, the rate of synthesis is 0. Right? So under conditions where the transcription factor is not present, we have a 0 rate. We might think of the reaction that we're undergoing in this case as the transcription factor has to be bound to the gene. And so the gene is bound to the transcription factor at its promoter, and that leads to the production of mRNA, and the gene and transcription factor unchanged in this reaction. Now, when we have a lot of transcription factor, remember the gene's a part of DNA. There's not an unlimited amount. When we have a large amount of the transcription factor, it will essentially always be stuck on the gene. We'll talk about this a little bit sort of more in a moment. And there will be a maximum rate. RNA polymerase can only move so fast. There's a finite number, often one copy of an individual gene. And there's a limit to how quickly you can push mRNA off of that. So when we have large amounts of transcription factor, mathematically, when the concentrations go to infinity or go to very large values, we reach some maximal synthesis rate that I'm also going to again use the symbol mu of x for. And in between these two extremes of 0 when we don't have transcription factor and mu x when we have a lot of transcription factor, we are going to use a function that varies smoothly from 0 to that maximal rate. And we can do this simply by multiplying the maximal rate by a scaling factor. And this scaling factor will go down to 0 when transcription factor is low, and up to 1 when the transcription factor is in high concentration. Right? What might this scaling factor look like? How might we come up with such a form? Well, here's where we're going to, again, be inspired by mass action kinetics to think about a little bit about mechanism and what might be going on. We may think of this psi x function, this as representing the fraction of the time that the transcription factor is actually bound to the gene, actually physically associated with the gene. Why might we do this? Well, we might think about the overall synthesis rate. Imagine that the gene can be in two states. The gene can either have the transcription factor present, or it can not have the transcription factor present. When it's not bound, the rate of the synthesis is 0. Nothing happens. When it is bound, produces mRNA at the maximal rate. The overall synthesis rate then comes from if the transcription factor is coming on and off, on and off, on and off, how often is it bound? If it's bound all the time or almost all the time, then the overall synthesis rate will be very close to that maximal rate. If the transcription factor is almost never bound, then the overall rate will be very close to zero. If the transcription factor is bound about half the time, then the overall transcriptional rate will be about half the maximal rate. Binding reactions, association is something that we have good models for based on mass action kinetics, and a very commonly used function that can describe a fraction bound is the so-called Hill equation. It was actually derived, originally developed in order to describe binding of oxygen to hemoglobin. So and it's precisely the fraction of the sites of hemoglobin that were occupied by oxygen as a function of oxygen pressure. It describes exactly the kind of phenomenon that we're interested in. How much of a component is bound to another molecule. It has all of the desired. This is the Hill equation shown here. It has all of the desired properties. When the transcription factor concentration is 0, we have 0 over a number plus 0, which is of course 0, has the correct low limit. When the transcription factor has high concentrations in the denominator, this term dominates. We can ignore this. We get a ratio of 1, so it goes to the right limit in the high transcription factor level, and the behavior of it is reasonable. We'll talk a little bit more about this in a moment. Given this expression for a fraction bound, for this psi as a function of transcription factor, we can then write down an overall RNA synthesis rate for an obligate activator given by the Hill equation multiplied by this maximal rate piece here. All right. So let's talk briefly about the Hill equation a little bit more. It can be derived from first principles, although there's many assumptions in doing so. It wasn't originally derived from first principles. It was originally actually fit to experimental data on hemoglobin, but afterwards, retrospectively, a derivation of it was found. And that derivation gives a very important meaning to this Hill coefficient, H. It's a measure of the degree of cooperativity of binding. Many molecular systems, many biomolecular systems have more than one place that a molecule can bind. And when that happens, apologies, sometimes the binding is independent, that the strength of binding one, the strength of binding another are the same. But in many cases, they're not. That after one molecule binds, it can make it easier for the next molecule to bind. This is exactly what happens in hemoglobin, for which the Hill equation was derived. There are 4 binding sites, and after the first one binds, it becomes easier for the second one to bind. After the second molecule of oxygen binds, it becomes easier for the third and fourth. And this makes a very, very highly cooperative system. What does that mean? It means that if you actually look at a population of hemoglobin has 4 places you can put an oxygen. Under moderate oxygen levels, you usually see some hemoglobin with all 4, and some hemoglobin with nothing, rather than a lot of hemoglobin with, say, one oxygen bound. Right? The same thing can happen with transcription factors. When there's more than one site on DNA that a transcription factor can bind, there can be cooperativity in that binding. And it's captured by this H. Apologies again, a little bit dry throat. All right. This degree of cooperativity can be loosely tied or related to the number of molecules that have to act together to have an effect. If there's 2 binding sites and there's strong cooperativity between them, you might expect a Hill coefficient close to 2. If there's 4 binding sites with high cooperativity, you might expect a Hill coefficient of close to 4. For hemoglobin, for example, the fit Hill coefficient is 3.7, 3.8, or something like that. It has 4 binding sites. This is only a loose relationship. However, as I said, hemoglobin has 4 binding sites. Its Hill coefficient isn't 4, it's close to 4. There absolutely can be cases where you might have 4, 5, 20 binding sites. And if you don't have extreme cooperativity, you may have a Hill coefficient of only 2 or 3, even though there are many more binding sites. There's a relationship, but it's a fairly loose one. There are some limits. If you only have 2 binding sites, you really can't expect to get a Hill coefficient anything more than 2. But if you have 10 binding sites, it certainly is possible to have a Hill coefficient much less than H. K half in the Hill equation is the concentration of half occupancy. If the concentration of X equals K half, then the 2 terms in the denominator are equal and they're double the value in the numerator. And so you get a value of a fractional occupancy of half. Right. You're going to explore the Hill equation a little bit more in the project. Okay, so our model, we've come up now with a model for obligate activation of a gene using the Hill coefficient, the idea of a fraction bound. With this framework, we're actually in a place to much more easily come up with a model for repression. Again, repression comes in many different flavors. In some cases, it simply reduces the rate of the reaction and doesn't take it all the way down to 0. In some cases, it can completely block transcription completely. In the simplest context, a repressor works in sort of exactly the opposite way of an obligate dimer, an obligate activator. When there is no transcription factor present, the gene can be transcribed at some rate of mu x. But when the transcription factor is present, when the transcription factor is present in excess and is bound to a high degree to the gene, it completely blocks the reaction. Transcription factors like these might be transcription factors that actually physically associate on the place where RNA polymerase needs to bind. And if that transcription factor is there, there's no way RNA polymerase can get in there in order to transcribe the gene. Right? So when there's no transcription factor, we have transcription. When there's a lot of transcription factor, we have the gene completely shut off, no transcription. Right? The same idea of fraction bound can be used to average this out. It just works in the opposite way. The contribution from bound state, the contribution from the bound state of the transcription factor is 0, and the contribution from the unbound state is mu x. And so what we really want to do to write the rate is we would like to write the rate as this overall maximal rate of mu x multiplied by the fraction that isn't bound. The fraction that isn't bound is the fraction that where transcription can happen. How do you get from a fraction bound to a fraction unbound? You simply subtract 1. Right? If 75% of the molecules are of the gene is bound, or the gene is bound sort of 75% of the time, then it must be unbound 25% of the time. If the transcription factor binding site is occupied 30% of the time, it must be unoccupied 70% of the time. And so all we need to do is 1 minus this same psi x, describing the fraction of the transcription factor bound, and multiplied by this overall rate to give a model for repressed transcription. So we've now gone through each of the individual pieces, degradation, translation, transcription, with modeling frameworks for each of these. How do we put it all together to actually write a model for the expression of a gene to a final functional protein form? Well, it's pretty straightforward. We're going to need 2 equations, one for protein, one for RNA. Remember, these are the 2 components we're tracking. Just as in the case for mass action kinetics, we're going to have differential equations, ordinary differential equations, a rate of change, a concentration over time. Both of our terms are going to have a positive term for synthesis and a negative term for degradation. The degradation term is going to be linear in the same species. And so for protein, the degradation term depends on the protein concentration. For RNA, the degradation term depends on the RNA concentration. Degradation is unimolecular in the same species. For synthesis, we have protein synthesis. Our simple model is the translation rate proportional to the amount of mRNA. And so we have mRNA in this term here. The example that I have here has a synthesis rate for mRNA that is just a constant. This is our model for a constitutively active gene. The schematic that I've written down here has gene X producing X mRNA, producing X protein with no regulation. A constitutively active gene that is just produced at some fundamental rate, which we can describe with this sort of model. Adding regulation into the picture, we actually can get regulation even when we only have a single gene. We can get this because some transcription factors actually affect their own expression. Some genes are autoactivators. Autoactivators turn themselves on in some sense. They activate their own transcription. That is, gene X produces X RNA, X mRNA produces X protein, and this protein can go back and can affect the transcription, can make it easier for the mRNA to be produced. How would we write such a model for this? Again, 2 equations, protein and mRNA. Synthesis and degradation for each. The degradation terms are exactly the same as in the previous case. Protein degradation dependent on protein concentration, RNA degradation dependent on RNA concentration. Protein synthesis, the translation term, is the same as in the previous model, dependent on the mRNA levels for gene X. However, the transcriptional rate, the RNA synthesis term, is different when we're under control regulation compared to when we're in constitutional activity. Here we have a model for the dependence of transcription on X protein. This is the model for an obligate activator. If there is no of that X protein, none will be produced. And if we take a look at this, in this particular model, we've put a Hill coefficient of 1, which is of course missing, but I could have written a 1 in each of these pieces here, which is the case for a non-cooperative mechanism, or maybe there's only a single transcription factor binding site, or the transcription factor and binding sites are independent from one another. And so we have no cooperativity that we need to describe in this. And this would be a pair of equations that describe the expression of that gene. There are some interesting things that you can learn about autoregulation. And again, autoregulation is also going to be something you explore in the first project. There is more that we can do with this. And this is where this area really becomes incredibly fascinating, and where models can tell us some very, very interesting behaviors that arise out of these types of regulatory structures, and really can provide insight into what's going on into biology. When transcription factors affect each other's expressions, we end up with gene regulatory networks of transcription factors that are all interconnected in some way. And there's all sorts of different ways that this can happen. The example we just described is an autoenhancer, a shorthand for writing out that central dogma-inspired cartoon from the previous slide is just to say protein A controls itself. We don't need more detail than this within our model, because we're only considering transcriptional control. And so if we say that gene A controls itself, we know that the way that, at least within the context of our model, the only way a gene can control itself is by that gene being a transcription factor that affects the transcription or the RNA synthesis of that piece. And so it suffices to have an arrow pointing at itself to say A activates itself. Other types of gene regulatory networks might include things like a transcription factor acting as some sort of a master switch. A single transcription factor is an activator for multiple different genes. So what this means is that at turning on or having the presence of one transcription factor has an effect that it turns on lots of things at once. This is like a switch on a home stereo system, that you flick one switch and it turns on multiple components all at once, so that you don't have to go through and turn on each one individually. There's a gene regulatory structure that can lead to that. We can have structures like this, a so-called negative feed forward, where gene A is an activator for gene B, and gene B is an activator for gene C, but A is also a repressor for gene C. This kind of structure can lead to delays in the response, right? So if A is activated, if A is sort of turned on in some sense, it will initially have an effect of holding down C, right? But even as it's holding down C, it is turning on B. And when B gets turned on, gets activated enough, reaches high enough levels, it can start overriding the repression of A to turn on C. And the result is that C, if some signal came in and turned in A, C would turn on a little bit later. It applies a delay in the response. Networks like this, I'm not sure if networks like this have been identified in natural systems, but there's a very interesting paper on an engineered circuit that has this feature of a triple negative feedback loop. A is a repressor for B, B is a repressor for C, and C is a repressor for A. This kind of structure can lead to oscillations in response. If you just walk through it, you might see why. If A is at a high level, then it will keep B as being low. But if B is low, B usually turns off C. So if B is low, then C will be high. But if C is high, it will turn off A. But we said A was high. So if A is high, it leads to A being low, right? We get this cycle, but there's going to be some delay in here because we have to move in from this. So we start off with A being high, and it leads back to A being low. Once A is low, the inhibition of B is turned off. That means B can grow. When B grows, it turns off C. When C is turned off, the repression of A is turned off. That means A can grow again. And so you end up with oscillations. Now, there's a lot of details into this to have actually prolonged oscillations, but it is possible to construct these kinds of networks. Right? These are actually what we call network motifs, little pieces of networks. If you actually look at the structure of how the transcription factors in the set of all transcription factors in an individual organism is structured, you will find that they often are, these networks are built up of many of these different kinds of modules linked in different ways. And our goal as modelers is to build a structure that allows us to look at these more complicated networks. How do we do that? Well, all we have to do is repeat the process that we did in those two earlier examples for each gene in the system. Right? Every gene in the system, we write a pair of equations, one for protein, one for RNA. And we use a common framework for each of these. For this triple negative feedback loop, the system of equations might be here. Sorry, there is a kind of important little typo that I just noticed here. These all should be negatives, not positives, because we need a synthesis term and a degradation term. With that correction, so for each of the protein terms, A, B, and C, each of them has a synthesis term, and the synthesis term is always dependent on the concentration of RNA for the same species. A protein is made from A mRNA, B protein is made from B mRNA, C protein is made from cmRNA. There's also degradation terms that depend on the same species. Degradation of A protein is dependent on A protein, and so on. The same form for each protein, although the constants may be different. Similarly, for the 3 species of mRNA, we have an equation that has a synthesis term and a degradation term. Again, the degradation terms all look the same. They all depend on the molecule of interest, A mRNA, B mRNA, cRNA. The degradation constants may be the same or they may be different. Then we have the mRNA synthesis piece, the transcription piece. Because each of these is repression, we have a similar form. The form for repression, if these were activation, we would need a different form. If one of them was constitutively active, we'd need a different form, and so on. But the differences really are limited to this transcription rate piece. And here, again, I've had a model that doesn't have any cooperativity, as I haven't put in a Hill coefficient there. We can do this as shown for the example for this triple negative feedback loop. Larger systems just require us to repeat the same process over and over. Although certain forms of control we haven't discussed how to model, but there's been a lot of work on how to do that. All right. In summary for this mini lecture, we've seen how translation, transcription, macromolecular degradation, complex processes that have a high level of many sort of pieces that go into making them happen, can all be modeled by equations inspired by mass action kinetics that allow us to really simplify down, abstract away the complexity of these processes into a manageable piece. They don't have to be simple rules, but they have a simple structure to them. In the simple models we've started looking at, the regulation is limited to transcription. We haven't put in any other kind of regulation. And with these simple models, we can easily build up models of complex gene regulatory networks simply by reproducing this basic structure for expression of a gene for each individual gene. And actually, what we find is that these very, very simple models can actually exhibit quite complex behavior once you start integrating them into these sorts of circuits. Okay, so that wraps up this particular mini lecture. And actually, this is the last scientific content lecture of the first module. I will be adding for this module some MATLAB-based mini lectures that describe how we actually implement these kinds of models in a computational framework so that we can start to learn about them and actually get some results out of them rather than just looking at a set of equations on the sheet of paper. Take care and be well.